Polymerase chain reaction, or PCR, is an assay of great importance in any molecular biology or clinical analysis laboratory. The procedure was conceived by Carey Mullis in 1986, what supposed an amazing methodological revolution, since it allows to amplify specific nucleotide sequences of nucleic acids in a matter of hours, in contrast to the long and boring cloning protocols formerly used. It is important to thoroughly control the volume amount and the composition of each sample by labeling the ependorf tubes that will be used for the PCR assay. Reactives must always remain on ice, regardless their magnesium chloride, primers, buffers, RNA, DNA or cDNA samples, usually in the same number and concentration, milk water or nucleotides aliquots. Polymerase enzyme should be at -20. In fact, it could be wise to avoid too many freezing and thawing cycles. When a PCR assay is performed, the inclusion of a control sample of a housekeeping gene is required to validate the results, as well as at least a positive and a negative control for the gene or genes of interest. The most of PCRs are performed within a range of volumes of 25 to 50 final microliters. So tiny volumes can only be handled with micropipetters. But recall, to take any volume, before putting the tip in the sample, the plunger has to be pushed just until the first block. Once the ependorf tubes are ready with their mix inside, it's recommendable to homogenize with a brief spin. It's advisable too to employ special tubes which avoid the nucleic acids to stick to their walls, because that could decrease the amount of DNA or RNA available for the reaction. The thermal cycle is a very useful tool where we can put our samples and schedule the temperature change and the cycles required to amplify DNA. The success of the assay will mainly depend on the design of the primer selected to bind the flank regions of the studied DNA sequence in the samples in a complementary way. Each PCR cycle is compounded by at least 3 stages, where temperature periodically changes. The length of this stage can also alter, depending on the kind of assay we perform. The first stage is the denaturation step, where double-strand helix of DNA is separated at 19.5 Celsius degrees. The second stage is the annealing step, where each primer, one per strand, binds its complementary sequence in the sample. This is usually carried out in a temperature range of 50 to 60 Celsius degrees, and it is directly related to the length and composition of the primers. Then, special DNA polymerase, which is able to work at high temperatures and not to be denatured, locates the primers and starts the replication of each strand DNA sequence using them as template by the addition of nucleotides. So, in the end, a new copy of the studied sequence is produced. In conclusion, this is the third stage of the reaction, which is named the Elongation Step, and it's performed at 72 Celsius degrees. The replication is always carried out in a 5 to 3 prime sense. Formerly, there was a single copy of the sequence, but now there are 2. Each cycle, the number of copies of the interest sequence is doubled. After 2 cycles, there will be 4 copies. After 3, 8. And after 35 cycles, 68,000 millions of copies for each sequence we got originally in the sample. While DNA amplifies, the second part of the assay should be prepared, the agarose gel. The concentration of agarose, which is obtained from some algae, will assess the density of the gel. With larger or smaller pores, the DNA molecules will go through in an electrophoresis procedure. By instance, if you wish to separate DNA fragments of similar size, you have to prepare a high-density gel, so a high agarose concentration will be required. With some foresight in a hood, we can prepare the tray, the combs, and the rest of tools we'll use next. The powder of agarose is added to a TAE buffer, and then it is warmed up until it is completely dissolved without lumps. Then it is poured in the tray, often inside the hood. The comb it is quickly put so it can shape the wells where the samples will be loaded. Before it solidifies, dangerous ethidium bromide must be added. What will let us visualize the DNA. Ethidium bromide intercalates the nucleotides. That's why it is carcinogenic, and why it should be handled in a hood, which avoids its toxic effect after inhalation. In this case, the homogenization of this dye inside the agarose mix is essential to achieve the same concentration of this reactive everywhere. When PCR is over, a new buffer is added to each sample to bestow them a higher density and to let us know we have properly loaded them. The solidified gel is put inside a tray with salt buffer, which must totally cover it to avoid it become overheated when current passes through it. The combs carefully move away so we get access to the wells. It helps placing under the tray a colorful element to better distinguish the wells. The loading of samples is a delicate step, since it is critical to avoid pricking the gel with the tip. The samples go down to the bottom of the well thanks to the higher density they have by the earlier addition of the load buffer, usually composed by glycerol and a dye. Now we put the lid and connect the electrodes to the tray and to an electric energy source. The DNA samples are negative charge due to their acid nature. What prompts they are repelled by the cathode and attracted by the anode. All the DNA molecules have the same charge density. What means their greater or lesser mobility through the gel only depends on their size. The yellow dye indicates the front of the electrophoresis reaction. We must end the procedure before no more gel to go through remains. Otherwise, DNA might be lost in the salt buffer. Once the electrophoresis tray is unplugged, we carefully take the gel where DNA molecules have been separated by size to a gel printer machine with a UVA lamp. When ethidium bromide, intercalated in the DNA, is exposed to UV light, it is excited and emits an orange light, which reports us where our amplified DNA sequence is located. Moreover, it must theoretically have the same length in every sample. If you want directly reveal the gel, it is required to protect your site with special goggles against UV light. Much more usual is to choose that the gel printer machine reveals itself, the gel, in an enclosed compartment, after what it shows us the capture image on the screen of a computer. This is an example of such image shown on the screen. In the first lane is included a DNA molecular weight marker, which must always be loaded in the gel with the rest of samples before the electrophoresis reaction starts. This marker is composed by DNA sequences of known size and let us know the size of the sequences amplified in the PCR. The smaller molecules have gone further in the job because of their lesser size and volume, whilst the larger ones have gone through a shorter way, because it's more complicated that they can advance in the agarose net. These PCR have produced several sequences with different size, what maybe points out primers have bound to different variables of the interest sequence they were designed for. Or perhaps that there are foreign sequences to the one we study, which share flank regions with the gene we have chosen to work with, what would bring bad news for us.